Hello everyone. Today we will discuss about the factors reducing the enzymal activity. Part 2. Part 1 is about the factors influencing the enzymal activity. Now first of all, the enzymal inhibition. In this we have further 6 subtopics, that is the competitive inhibition, non-competitive, uncompetitive, suicidal, allosteric and the feedback inhibition. Now first of all, the competitive inhibition. It is a reversible inhibition. Now as the name says competitive, so of course there will be competition between the particular substrate analog or the inhibitor and the particular substrate. Over here the inhibitor molecule actually competing with the normal substrate to bind with the particular enzyme, and over here the inhibitor is a structural analog, as because it is similar to the structure of the particular substrate, so it will compete with the particular substrate and will bind with the enzyme. Now just take this example. As you can see, succinate is actually converted into fumarate, okay, by the help of the this enzyme known as a succinate dehydrogenase. But in the presence of the particular malonate inhibitor, the competitive inhibition occurs and it will inhibit the succinyl dehydrogenase enzyme and will not form the product fumarate, because this malonate is actually structural analog of this succinate dehydrogenase enzyme. Now what happens is, since enzyme and inhibitor complex can react only to reform the enzyme and the inhibitor, the number of enzyme molecules available for enzyme substrate formation and particular after then the product formation is reduced. Suppose for an example, 100 molecule of substrate and 100 molecule of inhibitor are competing for 100 molecule of enzyme. So half of the enzyme molecule are trapped by the inhibitor and only half of the molecule are available for catalysis to form the product. Since effective concentration of enzyme is reduced, so of course velocity is reduced. Remember, because over here the amount of particular enzyme, free enzyme available for substrate will be less, because half of them is actually attached or binded to the substrate analog or the inhibitor. Now this is a very important example. Now remember, competitive inhibition is actually reversible. Why? Because when excess of substrate is actually added, so we can reduce the amount of enzyme inhibitor complex formation. For example, suppose we have taken 100 moles of enzyme and 100 moles of inhibitor, but we have taken a large amount of substrate, that is around 100 moles of substrate. So only 1/10 of the enzyme molecule are attached to the inhibitor and 90% are working with the substrate. So over here only 10% of the enzymes are actually binded with the inhibitor, while in the previous example I gave to you, over the 50% were binding, so 50% were used. But over here it is reversible and can be reversed by adding excess of substrate. Now we have to consider the graph. Over here this graph, this purple is the normal enzymal activity, okay, rate of reaction versus substrate concentration, so it is normal, okay. But over here as you can see, this competitive inhibitor actually reduces the rate of reaction as compared to the normal enzymal activity. And over here remember, the Km is increased but the Vmax is constant. I told you before that the velocity of the particular enzymal activity is reduced, okay, but Vmax, the maximum velocity, is remain constant as that of the normal enzymal activity, but the Km value is increased. Okay, now as because it is increased, the affinity for the particular enzyme for the substrate is actually decreased, because Km is inversely proportional to the affinity. Now the very important topic, the clinical aspect of the competitive inhibition. Now first of all, the sulfonamides. Now these sulfonamides are actually commonly employed antibacterial agent. Now you may know that the bacteria actually synthesize folic acid, okay, as they have to, because their main structural component in them is the DNA. So as because of this, for DNA formation they actually form the folic acid, and they mainly synthesize the folic acid by combining the PABA with the pteroyl glutamic acid. Okay, pteroyl glutamic acid is combined with the PABA to form the folic acid. While the sulfa drugs, this sulfonamide, this sulfa drugs is actually being structure analog to this PABA, having the same structure, as you can see, almost same structure as of the PABA. So it will competitively inhibit this PABA and will inhibit the formation of folic acid, and by this the bacteria will die. Now remember, the drug, these sulfonamides, can be taken easily and in a very normal amount by the human being body, as because this drug actually don't affect any kind of side effect in the particular human body. Why? Because folic acid is not synthesized by the human body, remember. Okay, we can take only supplement from outside, so there won't be any kind of inhibition or the prevention of the formation of folic acid in the human being body. So this drug is very widely used for as an antibacterial agent, because it will inhibit only the formation of folic acid in the bacteria and not in the human being body, because human being body cannot produce folic acid by itself. We have to take supplements from outside. And next is the methotrexate, have the structure 4-amino-entyl methyl folic acid, structure analog of folic acid, and so it competitively inhibit the folate reductase enzyme. As you can see in this figure, this folic acid actually is reduced by the folic acid reductase enzyme and will form the DHFA. Okay, but as the methotrexate will competitively inhibit this folic acid to combine with this enzyme, so of course DHFA is not formed, and for the THFA is not formed. Now this, as because the THFA is not formed, this will again inhibit the thymidylate synthase name enzyme. Now as because thymidylate synthase name enzyme is actually inhibited, that particular enzyme actually converts the deoxyuridine monophosphate into deoxythymidine monophosphate, and this deoxythymidine monophosphate is further converted into deoxythymidine triphosphate. Now as because this deoxythymidine triphosphate or DTTP is not produced, so DNA synthesis is inhibited in a particular cell. So now this DTTP, that means deoxythymidine triphosphate, is not formed, so DNA synthesis is actually inhibited, because DTTP is very important for DNA synthesis, and as because of this, cell division is inhibited, and so this particular methotrexate enzyme is used as anti-cancer drug. Now dicomerol actually structurally similar to the vitamin K, and so act as an anticoagulant by competitively inhibiting the vitamin K activity. Now the INH, isonicotinic acid hydrazide, it is commonly used as anti-tuberculosis drug and structurally similar to the pyridoxal, and prolonged use of INH will actually cause the pyridoxal phosphate deficiency. So this is mainly used as anti-tuberculosis drug, while this is mainly used as anti-cancer drug, and this is mainly used as antibacterial drug, and dicomerol mainly used as a anticoagulant. Now these are some of the examples of the clinical importance of the competitive inhibition. Now next is the non-competitive inhibition, also known as the mixed inhibition. It is actually an irreversible reaction. Now over here the poisons like iodoacetate, oxidizing agent, and the heavy metal ions like lead and mercury actually non-competitively inhibits the enzyme. Now what happens over here is, as you can see, this is the enzyme, this substrate, and this is inhibitor. Now over here the inhibitor is not structure analog of the substrate. As you can see, they both have a different structure, and so the inhibitor actually binds to a location different as that of the activation site or the binding site of the enzyme to the substrate, and so we can say that increase in the substrate concentration do not relieve the non-competitive inhibition. Why? Because the inhibitor is not the structural analog of substrate, and it can bind to any other site of the enzyme and can decrease the enzyme activity. Now the example of it are, first of all, the cyanide. It actually inhibits the cytochrome oxidase. Fluoride actually inhibits the magnesium, and so the enolase. Now you may know in the glycolysis pathway we have one particular reaction in which the 2-phosphoglycerate is converted into PEP, phosphenol pyruvate, by the enolase enzyme. Now this enolase enzyme needs magnesium, okay, for its particular activity. But if we inhibit this magnesium by the help of fluoride, this particular enolase won't act, and as because of this, the 2-PG is not converted into PEP, and further the glycolysis process will not occur. Now the iodoacetate actually inhibits the enzyme having the SS group in the active site. Now which actually have the SS group in the active site, that is the cysteine. So it will particularly inhibit the cysteine containing enzyme. Now last is the BAL, British anti-levisite dimer, is the caprol. It is actually used as an antidote for heavy metal poisoning. Now the graph of this, as you can see, this is the competitive graph and this is the non-competitive inhibitor graph. So as you can see over here, it forms a covalent bond, so irreversible process actually occurs. Why? Because the inhibitor actually combines with the enzyme by forming a covalent bond, and then the reaction becomes irreversible. The Vmax is reduced but the Km value is constant or unchanged. Why? Because the remaining enzyme molecule have the same affinity for the substrate. Now this is the comparison between the competitive and the non-competitive inhibition. Now the uncompetitive inhibition. Now what happens over here is the inhibitor does not have an affinity for binding with the free enzyme. Okay, as you can see, E as ES, right? This won't be binding with the inhibitor won't be binding with the free enzyme. So what happens? Inhibitor binds to the enzyme substrate complex but not to the free enzyme. And so in such cases both Vmax and Km are decreased. Vmax and Km are both decreased. Inhibition of placental alkyne phosphatase Regan isoenzyme by phenylalanine is an example of uncompetitive inhibition. So what happens? Free enzyme won't bind with the inhibitor, but the complex will actually bind with the inhibitor and will further inhibit the further reaction. This is the graph for it. Over here, as you can see, this is the uncompetitive inhibition and this is the graph without inhibitor. Now next is suicidal inhibition, also known as the mechanism based inactivation, is an irreversible reaction in which the inhibitor actually uses the enzyme's own reaction mechanism, okay, to inactivate it. That's why it is also known as the mechanism based inactivation. In this, the structure analog is actually converted to more effective inhibitor with the help of the enzyme to be inhibited. Okay, and the substrate like compound, that means the inhibitor, initially bind with the enzyme and the first few steps of it are actually done well or catalyzed. But after that, the new product formed by the inhibitor and the particular enzyme irreversibly bind with each other, and so the product won't be able to come out, and so no product is formed. So in simple words, what happen is the inhibitor actually binds with a particular enzyme and it will do the further process well, whole reaction actually catalyzed. Okay, but at the end what happens is this particular enzyme won't be able to form the product or release the product, as because this particular inhibitor actually very effectively binds with the particular enzyme and so product is not formed. The example of this is the inhibition of the ornithine decarboxylase name enzyme. Now what happens is the ornithine decarboxylase enzyme actually converts the ornithine into putrescine, and this for the putrescine will form the polyamine formation. The polyamine synthesis actually occurs by this putrescine. And remember, the polyamine synthesis is very important for the DNA synthesis. Now, in particular diseases like trypanosoma, the sleeping sickness disease, we don't need further bacteria and we have to kill the bacteria or the parasites. So we have to decrease the formation of the polyamine by reducing the amount of DNA in them. So what we will do, we will decrease the amount of polyamine, and for that we have to decrease the amount of synthesis of the putrescine, and so we will decrease the amount of ODC1 or inhibit this ODC1. So this ODC1 actually suicidally inhibited by the DFMO name inhibitor. This is the difluoromethyl ornithine. Okay, has been found very effective in the treatment of trypanosomiasis or the sleeping sickness. DFMO is initially inert, but on binding with the enzyme forms irreversible complex covalent bond with the coenzyme, and the amino acid residue of the enzyme is actually inhibited. In the mammalian cell, the turnover rate of ODC is very high, and so inhibition of DFMO is only transient because ornithine is very high. So only transient or temporary DFMO will act. So DFMO kills a parasite with no side effect to the patient, and so it is mainly used to treat the trypanosomiasis. The next example is the allopurinol. Now what happens is the hypoxanthine is actually converted to the xanthine by the enzyme name xanthine oxidase. Hypoxanthine to xanthine by xanthine oxidase. But what happens is this allopurinol actually will bind with this xanthine oxidase enzyme and will form the alloxanthine, and so no product is formed, and so there is strongly inhibition of the xanthine oxidase enzyme. Now last example is very important of the aspirin. Now what aspirin actually do? It will actually inhibit this cyclooxygenase enzyme by binding with the active site of this enzyme and acetylating the serine residue in the active center of the cyclooxygenase enzyme. Now as because this enzyme is now inhibited, there won't be formation of prostaglandin by the arachidonic acid, and this prostaglandin actually causes the inflammation, and so inflammation is subsided. Now next is the allosteric regulation. As the name says, allo means other. So means this allosteric enzyme actually have two sites, that is the catalytic site and the allosteric site. Now remember, at the catalytic site particular substrate actually binds, with the allosteric site allosteric substrate or other substrate can bind. Now the binding of the regulatory molecule can either enhance the activity of the enzyme, causing the allosteric activation, or inhibits the activity of the enzyme, known as the allosteric inhibition. Now if the particular regulatory molecule activates this particular allosteric enzyme, it is known as a positive modifier. That particular regulatory molecule is known as a positive modifier. While if the particular regulatory molecule actually inhibits or decreases the rate of reaction of the enzyme, it is known as a negative modifier. Now, binding of substrate to one subunit may cause or enhance the affinity of the other substrate to again bind to a particular enzyme. Okay, and more there is cooperativity occurring over there. So that is known as a positive cooperativity. If opposite thing occurs, it means binding of the substrate can cause the decrease in the further attachment or the affinity of the further substrate. It is known as the negative cooperativity. Now we have one term over here, this is key enzyme. What is key enzyme? The body uses the allosteric enzymes particularly for regulating the metabolic pathways. Such a regulatory enzyme in a particular pathway is known as a key enzyme and the rate limiting enzyme. The flow of the whole pathway is constrained as if there is a bottleneck at the level of the key enzyme, because at this particular key enzyme activity, if the key enzyme is present, then only the further reaction will go in the further direction or in the forward direction. If this key enzyme is not present, so what will happen? The further reaction won't occur. So this key enzyme is very important. Now the allosteric inhibition is more effective when substrate concentration is low. Okay, then only the allosteric inhibition actually occurs or is more effective. This is metabolically very significant. When more substrate molecule are available, there is less necessity for stringent regulation, because if we have more substrates, so of course there won't be use of key enzyme because the reaction will go well in the further direction. But if less, then we will use this key enzyme to do the further reaction in the forward direction. Now, very important example of this is phosphofructokinase enzyme. Now, what happens is in the glycolysis process, you should know that this phosphofructokinase is the rate limiting enzyme. If this is not present, then the particular glycolysis will not go in the forward direction. Now, we have over here two things, inhibitor and activator. Now, the allosteric inhibitors are ATP and citrate, while the activator over here are the AMP and fructose 2,6-biphosphate. So, more amount of fructose 2,6-biphosphate or AMP, more amount of further reaction will occur. Now, ATP is substrate for the reaction, but it binds to the allosteric site while acting as a negative modifier. The whole glycolytic pathway is regulated by the phosphofructokinase reaction only, and the energy charge in the cell is actually decided by the ATP by AMP ratio and decides the operation of the whole pathway. So remember, in short, we can say that in the key enzymes, we need this particular enzyme, okay, for this particular reaction to occur, and in that we have inhibitor and activator which can inhibit or activate this particular enzyme. Now over here the ATP actually inhibits this PFK1 by actually binding with the enzyme at the allosteric site and further inhibiting the process or the reaction. The other example are the inhibition of the aspartate transcarbamylase in pyrimidine nucleotide synthesis by the CTP end product, as well as the heme inhibiting the ALA synthesis reaction. Now, as you can see over here, this is a graph of this. Over here with the positive modifier, the particular reaction will go in the upward direction and further increment in the rate of reaction. While the negative modifier, it will be less. Over here the Km is increased and so the affinity is decreased and the Vmax is reduced. Now these are some of the examples of the allosteric inhibitors, activators and the particular enzyme. Now next is the feedback inhibition. Over here the term feedback inhibition means that the activity of the particular enzyme is inhibited by the product of the reaction. Now over here as you can see in the example, the glucose reacting with the ATP in the presence of hexokinase enzyme will form glucose 6-phosphate and ADP. Now if there is formation of a large amount of glucose 6-phosphate, so this will inhibit the hexokinase enzyme, and so for the G6P formation will be inhibited. Now we have one term over here, that is the end product inhibition. Now what happens is in this pathway as you can see, A actually in the presence of enzyme 1 will form in the B, then B into C and C into D. But when there will be more and more formation of the product, that means D, this D product will actually negatively inhibit or negative feedback will occur and it will inhibit the enzyme 1 and further the reaction won't occur to form the product D. Now next is the repression. Now till now we did the inhibition, now the repression. Now till now in the inhibition case, the inhibitor act on the enzyme directly. The inhibitory activity is noticed as soon as the inhibitor is added. But over here in the inhibition, there is no change or decrease in the amount of enzyme. The enzymes were same in number. Though they were binding to the particular inhibitor, the enzymes were same in number. But in the repression act at the gene level of the enzyme and the effect is noticeable only after a lag period of hours or the days, and the number of enzyme molecules is reduced in the presence of repression molecule. Now for example, let's take this example of the lag operon. In this we have regulator, promoter, operator and the structural gene. Now after passing through this hole, at the end the structural gene will form the ALA synthase name enzyme. Now this enzyme will produce the heme. Now if there will be a large amount of production of the heme occurred by the ALA synthase name enzyme, so this heme will actually act as a repressor or the co-repressor, and along with the aporepressor formed by the DNA from the RNA, they both will block the site of this operator, and as because this operator site is blocked, so further formation of the ALA synthase is inhibited and so further formation of heme is inhibited, and so over here the repression actually decreased by the decreasing amount of enzyme. Now last is the covalent modification. Over here the enzyme is either activated or inhibited by adding or removing a particular group from the enzyme, and so we can either activate the enzyme or we can inhibit the enzyme by particular removing or adding of the particular group. Now over here it is the zymogen activation or the pro-hormone activation. This is actually a reversible reaction in which we will either remove a particular group from the particular hormone or the enzyme, which will either cause activation or the inhibition, or either we will add one group which will cause a further hormonal, pro-hormonal activation or inhibition. Now common example of this is the protein phosphorylation. Now we all know enzyme actually formed by the protein, and in that particular protein we will add one phosphate group, okay, to activate that particular enzyme. And remember the phosphate actually added to the serine, threonine or the tyrosine residue of the particular protein. Now what happens over here is when the hormone binds to the membrane bound receptor, hormone receptor complex or HR complex is formed. The enzyme adenyl cycle is actually activated because of this HR complex. This activation is mediated through a G protein. Now the active adenyl cyclase actually converts the ATP to cyclic AMP, CAMP. Now this CAMP actually acts as, remember, secondary. This cyclic AMP actually acts as a secondary messenger, and it activates the protein kinase name enzyme by binding to the regulatory subunit. As you can see, it will bind with the regulatory subunits of this particular protein kinase enzyme. In the protein phosphorylation, the particular enzyme, the secondary messenger, what it will do? It will cause a phosphorylation of the particular enzyme, and so it will activate this particular enzyme. Now in some cases the receptor itself has tyrosine kinase activity, which is switched on when the hormone binds to the receptor, for example insulin. So this was all about the factors reducing the enzymal activity. So hope you understood well. Thank you so much.